we're going to see the vestibular hair cells. So here we have a cell of the vestibular system. Actually, it's more like something like this. This is a hair cell. This is the type 1, and here we have the type 2. So here we have a apical membrane. This will be the apical membrane, and all this is the basolateral membrane. On this side we have all this is the endolymph, and this is the perilymph. The composition of the perilymph is quite similar to the cerebrospinal fluid. Here we have a higher sodium than potassium. Higher sodium than potassium. So low potassium and high sodium. And here in the endolymph we have high potassium or kalium and low sodium or low natrium. So in the type 1 vestibular hair cells, the basolateral membrane, this has a calyx shape like this, and the nerve that would be around here, that is certainly one branch of the vestibular part of the vestibulocochlear nerve, is like this, also has this shape, and then continues like this. This is the dendritic process that is sensing this vestibular hair cell. As opposed to this one, this type 2, here we have many afferent and efferents, so axons and dendrites, but in a button shape. So this was for the basolateral membrane. In the apical membrane, we have the kinocilium, that is this long cilium, and here we have in descending magnitude like this sterobilia. So the kinocilium, because it's a cilia, or these are cilia, the larger one, they contain the classical structure of the cilium. You know, it is a pair of microtubules in the center, and then 9 pairs of microtubules. 1, 2, 3, 4, 5, 6, 7, 8, whatever it be. Actually, these are not completely microtubules because they are a little bit fused. But anyway, so this is the structure of this kinocilium. These are sterobilia that are microvillia that are made of actin filaments, but sometimes erroneously they are called sometimes stereocilia, even though they are not. They are stereobilia. And so each one of the cells has only one kinocilium and 50 or 30 stereovilli. Alright, so now between these ones we have links. These are called tip links. Now if the ciliums and microvilli move towards this side, that is towards the side of the kinocilium, then there is despolarization of the cell, and if they move in the opposite direction, it is repolarization or hyperpolarization. So first, even though the cells are in rest, they are a little bit active, but they will be more active if they are firing. So how does this work? Each one of these stereovilli has at most 2 channels. So here we have a lot of potassium. We have electric gradient and concentric gradient. So when the channels of potassium are open here, the potassium will enter to the cell and is a positive charge entering to the cell, so it produces this depolarization. Then the potassium will leak out through channels here on the basolateral membrane. And calcium, here we have calcium will enter into the cell and produces the releasing of neurotransmitters by diffusion of the vesicles here, and so there will be glutamate and aspartate being released here. So these are excitatory neurotransmitters and will lead to the synapse. Now, when these channels, so this happened when the stereovilli moves towards this side. Now, if the stereovilli move towards the contralateral side of the quiñoncilium, there is a closing of the potassium channels, and so the cell is hyperpolarized because now there is no potassium on the cell, and so it stops. So this is the mechanism by which or how the vestibular hair cells act. In the next video we will see a little more of the anatomy, the utricle, saccule, etc., the response to angular acceleration, linear acceleration, all that. But here I want to just talk about the vestibular hair cells. And they differ a little bit from the auditory hair cells. One of the differences is that the auditory hair cells lose the quiñoncilium